In this video, I am inviting you to a very important controlled study that proves homeopathic potentized forms of arsenicum album can antidote the biological effects of that drug in molecular forms. This research study indirectly vindicates the MIT explanation of high-dilution drugs in terms of molecular imprints or artificial binding pockets that can deactivate and antidote the pathogenic molecules that are conformationally similar to original drug molecules. This study was conducted by a team consisting of Philippe Bellon, Pathikrit Banerjee, Sandipan Chaki Chowdhury, Antara Banerjee, Sergio Jyoti Biswas, Susanta Roy Karmakar, Surajit Patak, Bibas Guha, Sagar Chatterjee, Nandini Bhattacharjee, Jayanta Kumar Das, and Anisa Raman Kutabukshadwaron Lab, 20 Rue de la Liberation, Sainte-Foy-les-Lyons, France, and Department of Zoology, University of Kalyani, Kalyani 741235, West Bengal, India, and published on December 26, 2005. Complete report is available at this link, http://www.nkbi.nlm.nih. Gov PMC Articles PMC 1375236. The team undertook a study to find out whether administration of potentized homeopathic remedy, arsenicum album, can alter anti-nuclear antibody titer in people living in high-risk arsenic-contaminated areas. But the weakness of this study was that they did not have any scientific hypothesis regarding the biological mechanism by which the highly diluted drugs, without any chance of original drug molecules contained in it, actually produce this biological effect. Even though this inherent weakness naturally led the researchers into wrong interpretations and conclusions, it no way reduces the historical relevance of this study in the scientific understanding of homeopathy. To examine whether elevated anti-nuclear antibody titers reported in random human population of arsenic-contaminated villages can be reverted to the normal range by administration of a potentized homeopathic drug, arsenicum album, randomly selected volunteers in 2 arsenic-contaminated villages and 1 arsenic-free village in West Bengal, India were periodically tested for their titer as well as various blood parameters in 2 types of experiments: placebo-controlled double-blind experiment for shorter duration and uncontrolled verum-fed experiment for longer duration.
Positive modulation of ANA titer was observed along with changes in certain relevant hematological parameters, namely total count of red blood cells and white blood cells, packed cell volume, hemoglobin content, erythrocyte sedimentation rate and blood sugar level, mostly within 2 months of drug administration. Thus, potentized arsenicum album was proved to have great potential for ameliorating arsenic-induced elevated ANA titer and other hematological toxicities. Both these controlled scientific studies have proved beyond doubt that potentized homeopathic medicines can antidote or reverse the biological effects of parent drugs. In the absence of original drug molecules, how could the homeopathic potencies exhibit such an action? The theory that potentized medicines mimic the parent drugs is obviously disproved through these experiments. If the active principles of high-dilution drugs were mimics of original drug molecules, they should have worked in the same way as original drugs. But these studies proved beyond any doubt that the action of potentized drug is in a direction exactly opposite to that of original drug. Only logical explanation we can provide for this phenomenon of opposite actions original drugs and their potentized forms lies in the molecular imprints of parent drug molecules being the active principles of potentized medicines. Molecular imprints can specifically bind to the parent molecules by their conformational affinity, and thereby antidote or reverse their biological effects. Indirectly, these scientific studies strongly support us in ratifying the molecular imprinting hypothesis proposed by MIT explanation of homeopathy regarding the molecular mechanism of homeopathic potentization and high-dilution therapeutics.